ahead and talk about Crotaline venom. So it might be easy to initially think of Crotaline venom as just one specific molecule or one type of substance. Unfortunately, that is just simply not the truth. In fact, Crotaline venom is a very complicated mixture of polypeptides, proteins, of which many of these proteins are enzymes. And some snakes have more of certain types of proteins and polypeptides and less of others, and so on and so forth. You get this mixing and matching. So what I want to do is I want to talk about the general categories of substances present in Crotaline venom. And these are your cytotoxic substances, your substances that can cause coagulopathies, your substances that can alter hemodynamics, and your substances associated with neurotoxicity. And I have asterisks there because I'm going to talk about this in more detail because it is a unique finding, and it is a finding that is more specific to two certain types of snakes that I'll talk about in a little more detail. And of course, we're talking about the crotalids, so we're talking about North American pit vipers. Okay, so let's talk about the cytotoxic or cell toxic substances. So within this group, and this is one that you definitely want to kind of put in the back of your mind because it comes up time and time again. And these are the metalloproteinases. Okay, you also have hyaluronadases, and you have beta-defensin peptides. So these two are enzymes, okay, and this isn't just a specific enzyme, this is a large class of enzymes known as the metalloproteinases. So these are enzymes that have some sort of metal. Zinc is a real common one in some of these proteinases. And so as you might guess, these are a large group of proteins or enzymes that are involved in proteins. They cleave molecules off of proteins, so they're going to change protein structure. The hyaluronidase, of course, is going to work with hyaline molecules, okay, cleaving things. And then you have these beta-defensin peptides as well. Now, all of these, the general pathophysiologic mechanism of action is they cause microvascular edema, okay? So you have in your capillary beds become permeable, they become damaged and they break down. This is done by compromising the basement membrane that holds cells together, okay? A compromise of the extracellular matrix around the cells themselves in the interstitial space, and that causes breakdown. Okay, and then that causes this fluid leakage, and there's edema, and that causes damage ultimately. And that in severe cases, this can also lead to myotoxicity. So you can get more localized cytotoxic effects. This is where you get the swelling, you get the blebs that can develop, and then you can also get underlying muscle damage or myotoxicity. And a secondary result, okay, so secondary problems that really concern us about these are, of course, going to be in the long term infection. Okay, you're going to have infection. And in the short term, because you're damaging muscle, you're releasing a lot of myoglobin, okay, you're going to be really concerned about rhabdomyolysis, okay, and that, of course, can lead to renal failure, all right? And if this was bad enough to where you developed compartment syndrome, particularly of a large muscle, for example, muscles in your legs, this might lead to limb loss, all right? And in severe cases, it might potentially lead to hyperkalemia, life-threatening hyperkalemia. So there are a whole laundry list of things associated with just these particular substances, these cytotoxic substances. But that's not the end of the story, because this venom can also contain coagulopathic, okay, substances that cause clotting disorders or coagulopathies, a whole list of those. So you've got these C-type lecithin-like proteins, or CLPs, phospholipases, specifically your A2 phospholipases, your serine proteases, and then a diverse group of molecules known as your disintegrins. And what do you know? Metalloproteinases just happen to be among the class of disintegrins. You have your small, medium, and large disintegrins, and then your largest are these metalloproteinases, which tend to be reasonably large proteins or enzymes. So your C-type lectin-like proteins, your CLPs, okay, these are primarily both pro and anticoagulopathic. So you can actually have pro-clotting effects. In some venom, particularly if the venom is very, very rich in the more pro-clotting, okay, types of substances, you may actually have a situation where you are clotting too much. And you can search YouTube and find a video where they put a drop of venom in a big cup of blood, and the blood just clots immediately. However, you also have anticoagulopathic effects. The phospholipases, okay, they primarily modulate how platelets work, okay, the thrombocytes. So this can lead to thrombocytopenia and ultimately can lead to platelet dysfunction, and that can cause anticoagulation. Okay, so that is going to make us more prone to bleeding. Okay, these serine proteases, they primarily cleave fibrinopeptide A and B from fibrin. And when you talk about this, and I'll just go into this in perhaps a little more detail. So when you have your fibrin clot at the end of your clotting cascade, okay, you have this really robust cross-linking that occurs, and you have a very robust clot that develops as a result of that cross-linking. And essentially what these serine proteases do is they break serine bonds, and they're highly specific. Okay, snake venom, crotaline venom, serine proteases are highly specific to this fibrinopeptide. Okay, the peptide bonding and the peptides that are involved in keeping this cross-linking intact, and that can come in and it can break up those peptides, okay, and make this cross-linked area much weaker, and this makes a clot, okay. It essentially makes a very weak clot, okay. It is a clot that when it forms, it tends to dissolve spontaneously and break down very easily, and it just is not a robust clot. Okay, the disintegrins, the most important one tends to be the metalloproteinases. So not only do we have metalloproteinases that can break down tissue, okay, can break down the integumentary system and muscles through myotoxicity, but also certain types of metalloproteinases can have primary antiplatelet effects. It can actually cause destruction of platelets, which leads to thrombocytopenia, which ultimately leads to anticoagulation. So the net, if you look at this in general, most of your substances tend to be much more anticoagulopathic or anti-fibrino. They tend to be fibrinolytic in nature. Okay, so moving up to hemodynamics. Okay, you can have alterations in your hemodynamics, and this is through substances like bradykinines and vascular endothelial growth factors within the crotalid venom. And this ultimately acts on blood vessels. It causes vasodilation. Some of this is through the release of histamines, particularly with bradykinines, because we know that these are very important pro-inflammatory mediating chemicals. So you can have direct vascular effects, vasodilation, which causes a distributive shock, and you can get hypotensive from that. And of course, if there's histamine release, histamine also has a vasodilatory mechanism associated with it as well. Okay, and then finally we have neurotoxicity. And again, the caveat to this was that I'm going to talk about this in more detail in a separate video. Of most notable interest when it comes to neurotoxicity are going to be these Mojave toxins and these Mojave-like toxins. And the two specific snakes that tend to be more problematic or more well known to have markedly more neurotoxic venom are, of course, the Mojave, okay, the Mojave rattlesnake, and the southern Pacific rattlesnake, both of which are known to have more neurotoxic venom. These are snakes that are very common to California. And some years ago, there was actually a show called Venom ER, where they chronicled some snake bites, and there were some notable cases that involved, I think, primarily southern Pacific rattlesnakes, where some of the patients had pretty significant neurotoxicities associated with those bites, and I believe one of the patients even died on this show. Now, like I said, not all snakes are going to have every single one of these and in the same exact levels or have the same exact actions. You may have some snakes with venom that is much more cytotoxic and coagulopathic, but not neurotoxic. Or perhaps you'll have a venom that has pronounced hemodynamic effects, but or hemodynamic and coagulopathic effects, but very little cytotoxic and neurotoxic effects. And you can kind of look at this as a smorgasbord, and you can kind of pull and mix and match things. And from an evolutionary biological standpoint, it looks like snakes actually evolve, and that is that the venom tends to match the prey animals where that particular snake is. And in fact, you could have like a Mojave rattlesnake in one area, and then just a couple miles away another Mojave rattlesnake, and perhaps there's a lizard that is very resilient to the neurotoxic venom, the Mojave toxin. I'm just making this up. I don't know if this is exactly what goes on. That you can, there's some papers that you can see, and I'm actually going to post some links to some abstracts so you can reference additional information if you guys want. But let's say that these lizards happen to be very resilient to the effects of the neurotoxic venom. Then perhaps the Mojave rattlesnakes in that area, even though they're known to have this Mojave toxin, perhaps their venom will be more cytotoxic and more coagulopathic, so they have a better chance at getting those lizards. Whereas, let's say I have another Mojave rattlesnake a couple miles away, and it evolved in an area where there are lots of rats and mice, and those particular mice, maybe a field mouse, is very resilient to cytotoxic and coagulopathic effects. And we actually see this with mice when it comes to a rat and mice killers with the Coumadin, that we've actually seen some mice develop some resistance to basically it's Coumadin that they use in a lot of the rat killers. And we find that some of these mice are very resilient to that, and we've actually had to develop so-called super Coumadin or super warfarin rat killers. And so I could see where rats, you could have an area where rats or mice were more resilient to these types of substances in the venom, and perhaps in those areas the Mojave rattlesnakes are going to have more neurotoxic venom. Okay, so that tends to be a function of evolution and fits really nicely into an evolutionary biology worldview, certainly when it comes to Crotaline venom. Okay, so I'm going to talk about the neurotoxicity in more detail because the mechanisms are very complex, and it does not involve sodium channels, and that's where a lot of our traditional neurotoxicities, when I think about neurotoxicity in toxicology, we think primarily about sodium channel blocking effects of certain medications like local anesthetics. That it does not seem to be the case, particularly with the Mojave toxin. That does not seem to be the case. It actually involves calcium channels, and that's why in the prior video I talked about the role of calcium and how it is important in the release of neurotransmitters, and that will form the foundation for understanding the what we believe is the general mechanism for Mojave toxin-associated neurotoxicity. Okay, guys, so this is a brief overview. of Crotaline venom and the basic pathophysiological effects of that venom on the victim. As always, thanks for hanging in there.